So I will talk about AFM and s-SNOM data processing. And so far, everything was a bit abstract and theoretical, so I thought we'd just do it live. And actually, this is not the first time that we're doing such a Gwyddion tutorial, but there's a really long, full version on our website, which is a few hours, but it's, I mean, you can speed it up in the boring parts, you can stop it in the difficult parts, and really follow along. So on the website, you can download some test data, so you will get exactly the same data that's in the webinar, and hopefully get to the same result. So my initial idea was I take this webinar, I make a short version of this here, but I realized it's not really useful because you can just watch it here. And also, Martin has made much more beautiful looking slides than me. So I deleted all my slides and I took the ones from Martin. The goal of this session is the steps that he mentioned we do now in Gwyddion, even though Martin said we shouldn't do it. This school is not only for teaching, it's also for discussing, right? So we can discuss why Gwyddion is good in some cases, why it's not so good in other cases. So the first problem that we can encounter are these phase jumps that we see here on the left side. And as Martin explained, this is just a problem of displaying the data. Whenever the phase seems to be smaller than 1 pi, it will jump up to 1 pi and continue here. And in the image, it looks like this. And then the reason you correct it, you need a substrate in every line of the image, and then you can subtract a plane. And you can write now a code in MATLAB to do this. Or I have brought the data example here. Maybe I plot first topography so you know what this data looks like. It should open very soon. Yeah, there it is. So we have here a topography image, and it's just a white square. This is a silicon dioxide square on the silicon substrate. So this is a sample that we're looking at. And we're looking at the optical phase. So in this example, I know that if we plot here, it's in a different color scale, we can see there's a really strong contrast and it's a really abrupt. So if we make a line profile from the blue area to the red, we can, we can really see that we have a super sharp jump here. It's from roughly -2 to +3. That's around 2 pi. And this is how you can identify there's something artificial going on. And the reason you can correct this in Gwyddion, and this is for the people from the Uweske summer school, I found a better way to do it. Actually, somebody in the webinar found a better way. I just copied it. Which is here this wrap value function. We click it. We say our phase is jumping between in an interval of 2 pi. We hit OK, and we're done. So, and this shows now why I like to use Gwyddion, because this was very easy and everybody can do it. The reason why Martin doesn't want to do it is that it's a bit dangerous. Because this will remove every 2 pi jump. And here we were lucky, there was only one jump, so it's very easy to correct. But if there's multiple ones, then maybe this algorithm is doing something strange. And it's not Gwyddion's responsibility to take care of this. It's your responsibility. And also, if I say now you can click this button, I'm also not responsible. You have to always double check. You have to always double check. And the way I would double check it here is, again, with the line profile. You can see it's a relatively smooth transition now. Maybe compare with literature. What do you expect? How high should this contrast be? And if this is all agreeing with each other, I think then it's okay to do this. Okay. So this was already the first example. If you have any questions, you can ask at any time. Otherwise, I will just continue with the next point. And this is how to correct for phase drift. And so here, the image, there's not really a strong drift, but if we zoom in to the substrate, we can see something like that. Yeah, so it's not homogeneous. And now what we can do, and I realized in the webinar it's not done in an ideal way. You can just remove a background from this image. And I want to remove just a plane in the vertical direction, so the slow scanning axis. And this is done, again, with 2 mouse clicks. We can subtract this. We have to readjust the color scale here. But what we can see, this already, it does not yield a good result. So even though the function is there, we have to pay attention. In this case, we are averaging over the whole image, and we are flattening a plane onto the whole image. So this is why we have to be a bit more careful. And this is what I like a lot about Gwyddion. You can just draw on the image which areas you want to use to fit this plane. So you can really just draw anywhere like this. You can draw. If you want to code this green area in MATLAB, it's a bit more difficult. And then you can make it arbitrarily complicated. If you look closely, there's a lot of dirt on the substrate. Do you want to include this in your fit, or do you want to exclude this? In Gwyddion, it's very easy to exclude this. To program, it's harder. Yeah. Okay, so now this is flat. So this is what you can do with Gwyddion. Let's go to the next traits, how to normalize images. And here it starts to become a little bit tricky with Gwyddion, because what Martin said, you should always normalize, or you should generally calculate with complex values, which is not possible in Gwyddion. Here we get luckily along. We can circumvent this, because if you write down the expressions you find to normalize, you just have to divide the amplitudes, and you have to subtract the phases. So it's all real values. In this case, we can still do it with Gwyddion. So let's do this. We can check maybe. We can open the amplitude of this. Yeah, here's the optical amplitude. And we can see already from the scale bar the values. It's some strange numbers where we don't know what they mean. What does an amplitude of 0.2 mean? It's unclear. So that's why we can again select an area, or maybe we can zoom into the substrate here. Here, the substrate. If we look closer, the substrate is also not homogeneous in this case. So we have some brighter areas like down here, and some darker areas like down here. Now the question is, which of these areas do you reference to? And I also don't have the answer, which is why we just average over all of this. Sorry. We just average over all of this. We want to know what is the mean value of this silicon substrate, which is 0.19. We can remember this value for later and just divide the whole image divided by this value. I know it's very fast how I click the buttons. The slow version is on the webinar. So this is now just divided every pixel of the image by the average value of the silicon. And now what we can see is the values here are around 1 on the substrate. We can even make a line profile. Let's do it like this, where we can see it's roughly 1 on the silicon. This is because I averaged. There's also dirt on the surface. This is included. So now we can say on the silicon dioxide, the amplitude is 0.7 times that of silicon. So this now has a physical meaning. This is why we normalize this. And in Gwyddion, yeah, it was easy. We can do the same with the face. I think I will skip this because there then finally what you will have to do, instead of dividing by the reference value, you subtract the reference value. Okay. So this is normalized. Has now a physical meaning, and yeah, we managed to circumvent the complex number problem. And now, this was the last part, and actually, this is something that applies mostly for plasmonics and for field mapping, polaritons. And this is something that I actually didn't work so much with. That's why I didn't really pay attention to it. But I noticed from Martin's talk it's really important, and it's really a limitation of Gwyddion. Sometimes you want to subtract a complex number from your optical images. And I don't know an easy way of doing it in Gwyddion. So for this type of data, I think you should use something else. At least if you want to subtract here this complex background. And now you can see I'm already at the end of all these slides. What I want to show is not much longer. It's only this one slide here, where I just compare different ways of processing data. So I talked now a lot about the Gwyddion. It's free, it's easy to use. Many functions are there, okay? I didn't show that many, like statistical analysis, different things. What I like a lot, and I think it's very helpful, is to work with the masks just by drawing on the image to include or exclude certain areas. These are the pros of Gwyddion, but we cannot use real-valued numbers, which I understand for some samples is a problem. In yellow, it's written, Gwyddion can be used for batch processing of a lot of images, and also it can be done in an automatic way if you use Python. So you it's not really user-friendly anymore. And on the other side, you have MATLAB, you have open source versions, and there are also free versions available, but it's not so easy to use. You have to write your own code. You have access to a lot of databases, which is great, but you have to implement them or install them, which is not great. For the masks, how to draw them, I'm not sure how you do that. Some programs can probably do this. But we can work with complex valued numbers, and automation is easy if you already have the script. And now, I think the conclusion of this comparison is I don't know of a software which can combine all the green points, but maybe there is one. If one of you knows a software that's easy to use as Gwyddion, but also has complex valued numbers, then please share, and yeah, I can have a look. And then maybe the next tutorial is on a different software. No, but for the polaritons, I mean, it would be good to have such a software. I'm just not aware of any. Yeah, yeah, exactly. Yeah, and with this, I'm already at the end. This is the summary slide. Then thank you, Martin, for the slides.
Thank you, Lars. Are there any questions? Yes.
Hi. So my question is, you choose, to remove your polynomial background, you choose vertical and of the order of 1. Mm-hmm. So any specific, how do you choose? Like, we should choose order 1 or 2, 1 or 3 or 5. So what is the criteria?
So the way I learned is you don't do any post-processing on your data unless you have a reason, mm-hmm. and you can explain why. And the goal is to do as little as possible, as little as needed. And for this slow scanning direction, we know it can take 10 minutes, 20 minutes for an image. Interferometer will drift. You have this gradient in that direction. Mm-hmm. And I assume it's a minimal processing, I just assume it's a linear drift. And I see if this is enough. In some cases, maybe you have a longer measurement, it's not linear anymore. Maybe you have to use higher order polynomials. Yeah, but for this, it's really important that you have a reference area where you can track what's going on. Like in this case, it was a silicon substrate. Yeah. So you can do more, but there should be a reason why.
Thank you.
Thank you for the presentation. I just have a comment maybe here that best would be not to choose one, but actually to be able to work both in Gwyddion and others, which in my case it's MATLAB, because it's really like you have some benefits here and some benefits there, and there is no a single software golden solution to fit the both. And one thing which is missing here, when you work with polaritrons, sometimes you need to do Fourier transform of the signal. And Gwyddion is not even close to that. There is this Fourier filtering, but to analyze Fourier spectrum, to find the k-vector and effective mode index, that's not really close. And do fitting, exponential fitting. So that's definitely something where you need to use some other softwares. But since here there are many students, my advice would be try to learn both.
Why not? It helps for sure.
Okay, no more questions? Yes?
And make it so a suggestion that to neaspec, it would be nice if you develop a software. Actually, it's a missing part of all your system. So just remove free there below. So make it not free and provide it with the system. So it's not free for everyone, but free for the neaspec users. So this would be a nice solution.
Very good feedback. Okay, then if there are no more questions, no more comments, one more? No? Then I think we are done for today, at least for the scientific tutorial sessions.